Inside a completely dark chamber, 4 living mice were placed beneath an extremely sensitive camera. The room contained no lamps, no glowing chemicals, and no visible source of light. Yet after the camera collected photons for long enough, the outlines of the animals slowly appeared. Their bodies were producing a ghostly glow. Then the researchers repeated the experiment after the mice had died. The light rapidly weakened. This was not body heat, radioactive radiation, or a digital effect. Living cells were releasing tiny amounts of visible light as chemical reactions unfolded inside them. Plants, bacteria, animals, and humans appear to do the same thing. Every living organism may be surrounded by a light signal so faint that our eyes never notice it. Scientists call this phenomenon ultraweak photon emission, sometimes shortened to UPE. It raises a deceptively simple question. Is this glow merely biological waste, like heat escaping from an engine, or does it carry useful information about what is happening inside living tissue? Ordinary bioluminescence is easy to recognize. Fireflies flash to attract mates. Some jellyfish glow when disturbed. Deep-sea animals use light to hunt, communicate, or disappear against the faint illumination above them. These organisms possess specialized molecules and enzymes that deliberately generate visible light. Ultraweak photon emission is different. It does not require a dedicated glowing organ. It is found in organisms that appear completely dark, including humans. Its intensity is commonly measured at only tens to thousands of photons per square centimeter each second. Under normal conditions, the surrounding light overwhelms it completely. Imagine trying to see a single candle from thousands of kilometers away while standing beneath the midday sun. The signal exists, but the background is far brighter. Detecting biological photon emission therefore requires light-proof chambers, long exposure times, and cameras capable of recording individual photons. The photons appear to emerge primarily from metabolism. Every living cell is constantly moving electrons, breaking molecules apart and building new ones. Much of this activity occurs inside mitochondria, the structures that extract usable energy from nutrients. These reactions are essential, but they are not perfectly controlled. Some oxygen molecules become highly reactive compounds known collectively as reactive oxygen species. At moderate levels, these molecules participate in normal cellular signaling. At high levels, they can damage proteins, membranes, and DNA. When reactive oxygen species interact with fats and proteins, they can leave certain molecules in excited energy states. An excited molecule cannot remain that way indefinitely. As it returns to a lower energy state, it can release the excess energy as a photon. The process resembles a tiny spark produced during oxidation. One event is insignificant, but billions of cells carrying out enormous numbers of chemical reactions can generate a measurable cloud of photons. This helps explain why living mice appeared brighter than dead ones. While the animals were alive, metabolism continuously produced excited molecules. After death, organized cellular activity declined, oxygen use changed, and the photon signal faded. The glow therefore seemed to track biological vitality rather than simply the physical presence of tissue. The same study also examined plants. Researchers injured leaves and exposed them to stressful conditions. The damaged areas emitted more photons than the undisturbed tissue around them. The light changed as the plant responded to injury, suggesting that the glow could provide a map of oxidative stress. To a human observer, the leaf still looked dark. To the photon-sensitive camera, the injury appeared as a bright region spreading through living tissue. That difference makes the phenomenon potentially useful. Doctors currently monitor biological processes using blood tests, dyes, radioactive tracers, biopsies, and imaging machines. Many of these techniques require physical contact, injected substances, or the removal of tissue. Ultraweak photon imaging could be different. In principle, a camera might observe chemical stress without touching the patient or adding a label. The body would provide its own signal. This does not mean hospitals are about to replace scanners with biophoton cameras. The light is extraordinarily weak, and its source is complicated. Temperature, circulation, metabolism, diet, time of day, and environmental exposure can all influence photon emission. Two bright regions might arise from entirely different biological processes. The challenge is not merely detecting light. It is learning what the pattern means. Researchers have already observed changes in ultraweak photon emission associated with inflammation, oxidative stress, wounds, and some diseased tissues. Cancer cells often have altered metabolism and unusually high oxidative activity, leading scientists to investigate whether their photon patterns differ from those of healthy cells. Some experiments have reported measurable differences, but this remains an early research field. A glowing region would not automatically identify a tumor. Researchers would need large clinical studies showing that the method can reliably distinguish disease from normal biological variation. The spectral information may be particularly important. Light consists of different wavelengths, which our eyes interpret as colors. Ultraweak photon emission can extend from ultraviolet wavelengths through visible light and into the near-infrared range. Different chemical reactions may produce different wavelength patterns. If scientists can connect those patterns with specific molecules or types of cellular damage, the glow could become more than a simple brightness map. It might reveal which chemical processes are active. Consider a mechanic listening to an engine. The presence of noise alone provides little information because every running engine makes sound. But an experienced listener can identify a damaged bearing or loose component from subtle differences in frequency and rhythm. Biophoton researchers are trying to develop the biological equivalent of that skill. They need to determine which wavelengths, intensities, and timing patterns correspond to particular cellular events. Human bodies also appear to emit photons in daily cycles. Earlier imaging experiments reported that emission can vary across the day and across different regions of the body. The face and hands, for example, do not necessarily glow at the same intensity or reach their peaks at the same time. This suggests that any medical application would need a careful baseline. A patient could not simply be photographed once and compared with a universal standard. Age, skin properties, recent activity, and circadian rhythm might all matter. The more controversial question is whether cells merely release these photons or actively use them. Light is an effective information carrier. Plants detect photons to control growth. Eyes convert photons into neural signals. Molecules inside cells can absorb light and change their behavior. It is therefore physically possible for one cell to emit a photon that is later absorbed by another molecule. But physical possibility is not proof of biological communication. For photons to function as a meaningful signaling system, cells would need to produce them in controlled patterns, direct or preserve the signal, and respond consistently when it arrives. Researchers would also need to show that blocking the photons changes biological behavior. So far, the strongest evidence supports ultraweak photon emission as a consequence of chemical reactions, especially oxidation. Claims that cells use it as a hidden optical communication network remain speculative. That distinction matters because the subject has attracted explanations involving consciousness, invisible energy fields, and other claims that go far beyond the available evidence. The existence of ultraweak biological light is real and measurable. Its proposed roles must still be tested individually. The phenomenon does not prove that humans possess a visible aura. The photons are too sparse for unaided human vision, and the color-filled images presented in research reports are usually processed representations of photon counts rather than photographs of what a person would see. Yet the measured reality is already remarkable without exaggeration. Life continuously converts chemical energy into motion, heat, electrical signals, and molecular structures. A tiny fraction also escapes as light. New cameras may make that light easier to study. Modern sensors have improved their ability to detect individual photons while reducing electronic noise. Better algorithms can reconstruct patterns from extremely limited data. Faster detectors may eventually allow researchers to watch photon emission change almost in real time instead of waiting through long exposures. Plants could become one of the first major applications. Crops experience drought, infection, and nutrient deficiencies before visible symptoms appear. If stress increases photon emission early, cameras might identify affected plants before leaves wilt or change color. A farmer could theoretically scan a greenhouse at night and see which plants were beginning to struggle. Researchers could test treatments without repeatedly cutting leaves or collecting chemical samples. Ultraweak photon measurements might also help evaluate food quality, seed viability, and environmental toxicity. Because the signal reflects active chemistry, it may reveal changes that ordinary photography misses. In medicine, the most realistic near-term use may be monitoring change rather than making a diagnosis from a single image. A clinician could compare the same wound over several days or observe whether a treatment reduces oxidative stress in a specific region. The patient would serve as their own baseline. Instead of asking whether one photon count is universally normal, doctors would ask whether the pattern is improving or worsening. Before that becomes practical, researchers must solve serious engineering problems. Cameras need dark conditions, and biological movement can blur the signal. Human tissue scatters and absorbs light, meaning photons produced deep inside the body may never reach the surface. Skin pigmentation and thickness could alter detection without changing the underlying metabolism. The images may therefore provide information mainly about superficial tissues unless new methods can reconstruct deeper sources. There is also a basic mystery at the center of the field. Scientists understand several chemical pathways capable of producing ultraweak light, but they do not yet know how much each pathway contributes inside a complete living organism. A mouse-shaped glow is the final output of countless reactions occurring in skin, blood vessels, immune cells, muscles, and internal organs. Turning that image into a detailed biological explanation will require experiments linking particular photons to particular molecules. The first photographs are not the final answer. They are more like the first X-ray image of a hand, revealing that an invisible signal contains structure worth exploring. More than a century ago, scientists discovered that the body constantly emits infrared radiation as heat. Today, thermal cameras can locate inflammation, monitor circulation, and help detect equipment or building problems. Ultraweak visible photons are far more difficult to measure, but they could eventually provide a different window into biology. For now, the glow remains at the border between established phenomenon and unexplored possibility. Researchers know living systems emit it. They know stress and injury can change it. They do not yet know whether it will become a reliable diagnostic tool or whether cells use it as information. Back inside the dark chamber, the mice were not shining like fireflies. Their glow was millions of times too weak for human eyes. But the camera recorded photons leaving their bodies one by one, creating an image from chemical reactions that normally remain hidden. Life does not merely consume energy and produce heat. It also leaks a faint trace of light into the darkness. The question now is whether scientists can learn to read it.